So today's session was, I think, very instructive. There were 3 talks. The first talk dealt mainly with mitochondrial dysfunction in a model of multiple sclerosis, EAE. Actually, they didn't use EAE, they used a transgenic system to actually create dysfunction in mitochondria to model what may happen in progressive multiple sclerosis, and I thought that was very insightful. The second talk was from the University of Michigan, Dr. Irani, who was speaking mainly about the cytokine biomarkers that they're developing for progressive multiple sclerosis, which I thought added to the inflammatory side of the story. And third talk was very interesting. It was from Phil DeJager, who has been responsible for organizing and analyzing genomic information, looking at genetic risk alleles, risk factors for multiple sclerosis, and maybe how that might tie into ultimately different phenotypes of multiple sclerosis. My own work focuses on B-cell antigen presentation, so something that wasn't necessarily part of those 3 talks, but we're using mainly an animal system, EAE, this experimental system for multiple sclerosis, to examine what features of B cells might participate in inflammation that drives progression of disease. I think a couple of questions actually highlighted the takeaways from the session. One person really emphasized in the question session that we have to really be careful about how we approach the analysis of the progressive forms of multiple sclerosis. In other words, are we really analyzing the right cohorts of patients, and are we actually including the correct controls? And that goes across the board, whether we're looking at mitochondria or cytokines or the genes. So who we're including in the trials are very, I think, is extremely important question. So the important parts of the talks were that we have an appreciation maybe on the neuronal side of what could go wrong, which might drive progression. We have an assessment or appreciation of sort of maybe immunologic markers, so how we could maybe predict or stratify patients, so on the immunologic side, and of course the genetics is extremely interesting. But one of the concerns that I have about that session was really there's very little overlap yet. We really haven't blended these things together, and sort of at the end of the session there were some talks, sort of conversation about the fact that perhaps maybe remyelination or some of the facts in healing or recovery from disease burden could be the overlap between those 3 things. I think a lot of what was focused on is just progressive MS, but we do have those distinctions, primary versus secondary. And again, the distinction matters. I think that's one thing that we can take away from today's session. I think that's sort of an obvious statement, but moving forward, we really need to pay close attention to the delineation between primary versus secondary progressive disease. That wasn't really highlighted as much today. How to go about approaching that may be, I think, a little bit beyond what I can offer you today, but the idea that we can identify that there's a distinction there is extremely critical. As well, and there's a session now that's sort of talking about the pathology of MS, we really appreciate that maybe splitting rather than lumping is an important concept for us so that we can define specifically what falls under the umbrella of multiple sclerosis. And I would allude to, for example, the discussions that took place yesterday about neuromyelitis optica and the antibody-mediated features of that disease. How we might be able to learn something about the progression of NMO could really parlay into understanding how we identify, distinguish, and maybe ultimately treat progressive forms of MS, primary as well as secondary. It's been a long struggle. Of those 14, I think one of them is Novantrome for progressive multiple sclerosis, which we, at least myself and many of my colleagues, tend not to use very much because it's highly toxic and can only show moderate or mild benefit, if that. Right, so where are we headed? In what direction do we need to head to get to those therapies is a good question. I'm not sure today or at this meeting we're going to actually develop that. But I think, again, where we start to perhaps identify the targets. Dr. Irani's talk on the cytokines, for example, from the University of Michigan says, well, if these are biomarkers, or if we can say that these are distinguishing the different categories, progressive versus relapsing MS, that doesn't necessarily tell us about cause and effect, but it may prompt us to perform a trial and to ask whether or not, if we target those specific cytokines that are seen elevated in progressive MS, can we make it better or prevent the progression? Yeah, I think that's actually a legitimate concept. So how do we get to that point is really, I think, beyond this meeting, unfortunately. I'm a little bit biased. I study B cells. I feel that there is an extremely important role for them. Identifying what that role is can be quite challenging. So I think of B cell contributions to pathogenesis, sort of the development and propagation of MS, as falling into three camps. One would be their dialogue with T cells. I believe that's actually the most important feature, since we've long held that T cells are the centerpiece of the pathology and pathogenesis of MS. Another component would be antibody production. Now that, of course, would imply that there's some dialogue with the T cell to begin to have more specific and high affinity antibodies that are made. But again, the B cell is capable of producing antibodies that could be pathogenic. The third would be cytokine production. And yesterday's session was very interesting, the CME program, in which Dr. Amit Bar-Or was alluding to the fact that perhaps there's a bystander effect, that B cells, whether they are targeting a specific portion of the nervous system, within the CNS, within the central nervous system, they could still be exerting some inflammatory and destructive effect. So those three things sort of, I believe, all together, and we have mechanisms for each of those three items that B cells contribute to as potentially contributing to the pathogenesis of multiple sclerosis. I believe that those three actually substantiate that there's a big role for B cells. On top of that, we actually have very good data that if you deplete B cells, that's highly effective for at least relapsing or remitting multiple sclerosis, and apparently it may be effective for progressive MS. We've got a long way to go. One of the things that I'm particularly interested in is localization, compartmentalization of B cells. When B cells traffic to the CNS, I believe there's actually a waylay in the meninges or some interface at the barrier, and at that particular site we may be actually seeing some of those pathogenic effects being exerted. For example, follicular organization of B cells with T cells, and immediate or proximity to areas of damage within the cortex. And so those are kinds of things that I think about a lot.